It's a very exciting new seminar format that we have here today. It's to highlight work from awardees of the network's Health Informatics and Data Science competitions. We'll be hearing from awardees from the first 2 rounds of competition. They were awarded in 2023 and 2024. The 2025 awardees, we may schedule a new series in the fall. And these awards were 1-year projects meant to use network data to advance high-quality precision oncology research. The award is designed to support young researchers during their graduate and postdoctoral studies. And some of the speakers you'll hear from today have completed their studies and launched their careers since the award was ended, which is really, really exciting. We have now a new round of awards open at the moment, so please go to our website and check the details. It's open until April 9. And also, I want to highlight that we are accompanied by members of the network patient working group. We also have researchers, clinicians, and trainees, and leadership from all network consortia. And we are also joined by some of our funding partners, including staff from the Terry Fox Foundation. So I'm going to pass it over to Veronique Leblanc, who has been spearheading these efforts with the seminars and doing a fantastic job. Veronique?
Thank you so much, Isabel, and welcome everyone. As Isabel mentioned, this is a little bit different from most of the other seminars that we hold within the MOHCCN. And we've asked presenters here to prepare 3-minute presentations using a single slide to share their work in plain language. So it's a little bit similar to the 3-minute thesis competition, although we won't be doing any judging today. I also want to note that a unique aspect of this series is that it actually involves an element of training in and of itself. About 2 weeks ago, the presenters you'll hear from today met with volunteers from the network's patient working group to practice their presentation. And they received really thoughtful and constructive feedback from both patient working group members who were there as volunteers as well as their own peers. And we think that this is a really great opportunity to bring network trainees or former trainees together with people with lived experience to learn from each other and to help early career researchers develop their skills in sharing their research with a broad audience. So a big thank you to everyone who participated in that, especially Sally, Jennifer, and Kirsten, our patient working group volunteers. I'll also start off with just a little bit of housekeeping. We will be keeping questions until the end, so we'll do all of the presentations first. Please either type your questions in the Q&A box, which you can find at the top of your screen. It's 2 little speech bubbles with a question mark. Or once we get to the question period at the end of all the presentations, you can raise your hand, which you can also find at the top of your screen, and then we'll allow you to unmute yourself and you can ask your question live. I also want to take a moment to acknowledge that I personally am calling in today from Mi'kma'ki, the traditional and unceded territory of the Mi'kmaq people. Our network members and our speakers and our attendees today are located all across the country on lands that are home to diverse First Nations, Métis, and Inuit populations. And I invite everyone here today to honor and pay respects to those peoples and to reflect on the importance of those lands and waters that we all call home. Finally, I have the great pleasure of introducing my co-chairs, Sally and Jennifer. Sally and Jen both joined the network's patient working group in 2024, and they've been involved in so many activities in various aspects of the network since then. On top of all of this that they're already doing, they generously volunteered to, as I said, attend the practice sessions a couple of weeks ago with the speakers to give them feedback on their presentations and also to co-chair the seminar with me. So thank you so much for joining us, Sally and Jen. I'll pass it to Sally first if you want to tell us a bit about yourself and get us going.
Thanks, Veronique and Isabel. I live in Winnipeg, Manitoba, on the original lands of the Anishinaabe, Cree, Oji-Cree, Dakota, and Dene peoples, Treaty 1 territory, and the national homeland of the Red River Métis. I appreciate the spirit of reconciliation and collaboration with Indigenous communities. Diagnosed in 2021 with stage 3C melanoma, my treatment was informed by precision oncology. Because of improved treatments, I am a survivor with a very good quality of life and with no evidence of disease currently. In learning about how standard of care had been improved, I also learned about the tremendous work of the network and heard about an opportunity for me to give back. It was then that I applied and joined the patient working group. During these 2026 Winter Olympics, It's very exciting to be able to introduce the Team Canada of Cancer Research, the members who will be presenting their work today. One very impressive thing about these team members is how they support and encourage each other. Terry's mission is in good hands. And now Jen will say a few words.
Thank you so much, Sally. I would also like to first start off by respectfully acknowledging the territory in which I gather virtually. As the ancestral unceded homelands of the Beothuk and the island of Newfoundland as the ancestral unceded homelands of the Mi'kmaq, Beothuk, Innu, and Inuit. It's by learning our history that we can do better for the future in a spirit of truth and reconciliation. For most of my adult career, I've worked in some area of medicine. Never did I think that the tables would turn and I would become a patient. In 2019, 8 weeks before I was set to walk down the aisle, I was diagnosed with an aggressive form of melanoma that was already staged at 3B. Luckily for me, and for so many other patients, just 2 years prior, due to research from early researchers like the ones we will hear from today, a new immunotherapy drug was available for treatment. Before this, my cancer would have been considered a death sentence. 3 surgeries, 2 reoccurrences, and 1 year of immunotherapy later, I have no active signs of the disease since 2021. This is why I became part of the Marathon of Hope as a patient partner. I love the fact that I have the opportunity in our practice sessions and in these seminars to help researchers have access to people who have had lived experience outside of a lab setting. This allows me to do my part in making Terry's dream a reality of one day no one will have to face this terrible disease. These 8 presenters that you will hear from today are all recipients of the Network's Health Informatics and Data Science Awards. Their innovative research is helping advance precision medicine for all Canadians, and these training awards are helping prepare the awardees for what is sure to be a successful career in this field. And now, with no further ado, our first speaker today is going to be David Henshoff. He was awarded the Time of Award, a postdoctoral research fellow in Robert Grant's lab at Princess Margaret Cancer Center. David will be telling us about his work using machine learning to study pancreatic cancer.
Thank you very much for the opportunity to present today the work that I've been doing on using artificial intelligence to improve prediction on pancreas cancer patients. So on the left of your screen, you can see the clinical course of a patient with pancreas cancer. They receive a diagnosis, some systemic therapy, and some form of tissue procurement, which is a biopsy or surgical resection. All of these give different modalities of data to inform us on how to treat the patient. We have clinical data, we have imaging, we have histopathology, which is microscopic analysis, and the genome, which is the blueprint of the cancer cells. We studied whether each and every one of these data modalities could tell us regarding the outcome of the patients. And then we created an artificial intelligence algorithm fusing all of these data modalities to see if it would improve prediction. On the top right of the screen, you can see that we used 2 cohorts of pancreas cancer in randomized controlled trials on chemotherapy. One cohort about 200 patients for training and another one about 200 patients for validating our algorithm. And what we see from the right of the screen to the left is that each data modality contributes some information, but it is the late fusion, so the fusion of all of the modalities that give us the most accurate prediction. Then finally, on the bottom panel, you can see survival curves. The patients are classified according to the PURIS classification, which is a classification of pancreas cancer that is well known in research. And then you see that there are 4 curves because when we apply our algorithm of machine learning and artificial intelligence, we can refine the prediction better than what is currently the standard of care in research for pancreas cancer. So our algorithm was more powerful and more accurate. The future goals will be to validate this in external cohorts, and hopefully this will create a tool to inform clinicians on how to better improve the care of pancreas cancer patients. Thank you very much.
Wow, thank you, David. Our next presenter, Emma Rousselle, is a PhD candidate in April Rose's lab at McGill University. She'll talk to us today about her work in colorectal cancer.
Hello, everyone. So first, I wanted to thank the MOH for funding this project and giving me the opportunity to tell you about it. So BRAF mutations make up 15% of colorectal cancer cases, and there's an important distinction between the classical V600 mutation and the non-V600 mutation, and it's the requirement for another protein in the non-V600s for the mutant BRAF to have an effect and lead to tumor growth. And unfortunately, there are no approved therapeutics for the non-V600 mutant colorectal cancers. And so we wanted to ask, what are the differences in these two types of mutations? So I analyzed publicly available data and found that these non-V600 mutations were more prevalent in males. They were more likely to be left-sided tumors and occurred in patients younger than 60 years old. Now, these are important characteristics for physicians to be aware of since these cancers behave differently and should be treated differently. And from this database, I also analyzed other mutations present in these cancers and found that the V600 colorectal tumors had higher tumor mutation burden compared to the non-V600s. I looked deeper at the primary and metastatic tumors and looked for other genes that were mutated. And in the non-V600 tumors, as you can see with the dot plot on the bottom there was a clear preference for genes that were mutated in the same pathway as the BRAF gene, which would suggest that these tumors heavily rely on this pathway to grow. I also analyzed the transcriptome of these publicly available tumors, and this would provide information about the behavior of the tumors themselves. And I looked for gene signatures that were significantly different between the V600s and the non-V600s. Now, gene signatures encompass a set of genes that have similar functions, and the function that piqued my interest was the interferon gamma activity. This typically activates an immune response pathway that has a role in regulating inflammation. I performed other analyses which also suggested that there are different types of immune cells in the V600 versus the non-V600 colorectal tumors. And so we believe that the V600 colorectal tumors have an immunologically hot environment, which means that there are many immune cells surrounding or infiltrating the tumor when compared to the non-V600s. And so we hypothesized that patients with immune hot tumors would better respond to immunotherapy treatments versus the non-V600 tumors, which show that they would be better candidates for targeted therapies as their mutational background is heavily reliant on the same pathway as the BRAF mutation. So this work emphasizes the impact of different mutations in the same genes and how these cancers should be treated differently. Thank you for listening.
Thank you so much, Emma. That was very informative. The next presenter that we have is going to be Yuanchang Fang. At the time of the award, he was a PhD candidate in Faiyaz Notta lab at the University of Toronto and the Prince Margaret Cancer Centre. Today, he'll tell us about his work using liquid biopsies to characterize pancreatic cancers.
Thank you so much, Jen, for your kind introduction. So today my talk will be about the plasma whole genome sequencing to monitor pancreatic cancer. So pancreatic cancer is one of a very lethal malignancies. The 5-year survival rate of this disease dramatically decreased from 20% in the early stage resectable tumors to only 3% in the very late metastatic cases. So let's suggest that we need a very sensitive technology to identify this disease earlier so that we can treat the patients earlier to prolong the survival. Liquid biopsy is one of the most popular technologies that we use to identify the tumor signals from the patients. So when tumor cells die, they release DNA fragments into the blood circulation, and these DNA fragments carry mutations. We call them circulating tumor DNA. By conducting the sequencing of the DNA fragments from patients' blood, we will be able to identify those ctDNA or circulating tumor DNA from the patients so that we can identify the tumor signals from the patient. To validate this technology, we established a study cohort of blood plasma from pancreatic cancer patients, and then we conducted plasma whole genome sequencing. So on the top right, I want to show you some of the data. Basically, on the y-axis is the plasma ctDNA level. So basically, that means that how much of the tumor signal you have in the blood. And then clearly we can see the ctDNA level dramatically increase from the early stage resectable cases to very high in the metastatic cases, suggesting that the ctDNA level aligns with the tumor burden in the patients. And then finally, I want to show you a case study where we have a pancreatic cancer patient who was initially diagnosed with a primary pancreatic cancer. And then the patient received a surgery to remove the primary tumor. So 3 days after the resection, we obtained the blood, and then we measured the ctDNA level in the blood. So interestingly, we found that the ctDNA level was about 2%, which means that even though we removed the tumor from the patients, but there was still some tumor signal in the patient after the resection. So unfortunately, 70 days later, the patient got a relapse in the liver. So basically, all this data suggests that when the tumor size is very small, it couldn't be detected by the CT scan imaging, but we will be able to use plasma ctDNA to identify the disease earlier. So that eventually we will be able to treat the patients earlier. Thank you so much for your attention.
Thank you, Yuan. Some interesting, exciting advances for patients. Next up is Andrew Garvin, a PhD candidate in Amber Simpson's lab at Queen's University, who will talk to us about his work with bladder cancer.
Great, thank you so much for that introduction, Sally. So bladder cancer is very treatable for most patients at early stages. These tumors often respond well to standard therapy, and these patients have very acceptable survival outcomes. But for about 1 in 5 patients, something changes. The cancer suddenly becomes aggressive, it invades the muscle and local tissues, and as a result, survival probability drops dramatically. And we don't fully understand why this happens. One clue is something called cell identity. Healthy bladder cells in many early cancers have a very clear identity. They behave like normal bladder tissue. But aggressive tumors, they seem to forget who they are. They lose their specialized features, and they revert to a more primitive stem cell-like state that allows them to grow faster and spread more easily. We call this plasticity, the ability for these cells to change their identity. And my research question simply asked, what pushes bladder cancer cells to make this dangerous switch? At the top of the slide, you can see one stress that tumors commonly face: low oxygen. As oxygen drops, cancer cells from other cancer types have been shown to start a shift from this less aggressive state to a more aggressive one. And so we wondered whether low oxygen might actually help drive this change in bladder cancer. So first, we looked at hundreds of real patients in a large public cancer database, and we focused on 2 things. One, a common genetic mutation called ARID1A loss, and low oxygen within the tumor microenvironment. And surprisingly, one alone actually didn't affect survival very much. But when both happened together, the outcomes changed dramatically. About half as many patients survived at 5 years, and those tumors predominantly presented with a more aggressive cell identity. Next, we looked directly at patient tumor tissues under a microscope, and we stained them for signals of cell identity and oxygen deprivation within those tumor samples. And these are tumor samples that are in the midst of making this aggressive cell identity transition. And really, we saw the same story. The most aggressive cells were physically located in low oxygen tumor regions. Finally, we tested this in the lab. We simply exposed bladder cancer cells with this less aggressive identity to low oxygen conditions. And surprisingly, or maybe unsurprisingly, these cells changed their identity. They became more aggressive as a result. And so across patient data, tissue samples, and lab experiments, we really saw the same pattern. Low oxygen combined with certain mutations, particularly in ARID1A, actually helps tumors lose their identity and become more dangerous. So if we can detect or block this process early, we may be able to predict which cancers will progress and intervene before they become life-threatening. And that could mean better outcomes and longer lives for bladder cancer patients. So thank you so much for your attention.
Thank you so much, Andrew. That was really eye-opening work. Our next presenter today is Julia Negrin. She's a PhD candidate in Benjamin Haddy's CAD's lab in the Department of Medical Biophysics at the University of Toronto. Today, Julia will talk to us about her work identifying new therapeutic options for cancer patients.
Thank you for the introduction, Jennifer. Hello, everyone. Today, I'm really excited to share some results from my investigation into the potential of synthetic lethality for guiding precision oncology. This is an approach that promises the selective targeting of cancer cells while minimizing harm to healthy cells. Now, a really easy way to understand synthetic lethality is to think of skydiving. So skydivers, they have two parachutes so that if one happens to be defective or broken, the second one can be deployed. And this idea extends to our cells. So for some pairs of genes called synthetic lethal gene pairs, if one gene happens to be broken, cells can rely on the second gene to continue functioning as usual, but only if both genes are broken, so if both parachutes are broken, then cell death occurs. And as we know, mutations are very common in cancer, and so many cancer cells already have a broken gene or a broken parachute. So looking at the first figure on the left, if we know the first gene is only broken in cancer cells, then if we break the second gene, that second parachute, with, for example, a targeted drug, then these cancer cells can be selectively killed via synthetic lethality. Meanwhile, our normal healthy cells, which had the two normal genes to begin with, they still have one normal gene, and hence they continue functioning as usual. So here what we did, we developed a computational framework to identify and detect these gene pairs, and we applied this framework to 8 MOHCCN cohorts. Our results show that approximately 76% of all patients had at least one druggable synthetic lethal gene pair, meaning there was potential to induce synthetic lethality within their tumor cells. So to see what this means for cancer treatment, I want to walk through a real example of a gene pair from our findings. We identified this pair, you can see in the middle, involving a gene called KRAS, which is commonly broken in cancer cells, and a second gene called BTK. So just like in our skydiving analogy, cancer cells with a broken KRAS gene are already missing one parachute. So if we use a drug to take out the BTK gene, which is the second parachute, then these cancer cells should die while our healthy cells should remain unharmed. And that's exactly what we observed when we tested a drug called ibrutinib, which targets the BTK gene. So the two purple box plots labeled 1 and 2 there on the left are cancer cells with a broken KRAS gene, and we see they respond strongly to this drug. The two brown box plots on the right, labeled 3 and 4, are cells with a normal KRAS gene, and these are largely unaffected, just as our framework predicted. And so to summarize, here we demonstrate two things. First, that these targetable gene pairs are widespread across diverse patient populations. And second, by identifying these pairs, we could better match patients to treatments that are more likely to be effective to their unique cancer cells while reducing unnecessary adverse treatment effects. Thank you very much for your attention.
Thank you, Julia. Minimizing harm is good. Our next presenter is Jorge Pinzon Mejia, a PhD candidate in Jeanette Boudreau's lab at Dalhousie University. He'll be talking to us about his work in colorectal cancer.
Thank you for the introduction. So colorectal cancer is the fourth most common cancer in Canada, and it accounts for the second highest amount of deaths. And more importantly for us, it has the highest incidence in the Atlantic provinces, so it hits really close to home for us in Nova Scotia, and that's why we are very interested in researching it. One important to consider is that patients within the same stage of the disease can have very different responses to therapy. Some patients won't respond to the same drug, and also they can have very different outcomes. So some people can go and live for a very long time, while others will succumb very quickly to this disease. And that's why we need better understanding of the disease itself, of how our body is reacting to it, mainly the immune system, in order to get better classification systems and also in the hopes of developing better tailored treatments. So our research group focuses on understanding what the immune system is doing in the presence of a tumor. And for this, we research patients with stage 3 colorectal cancer. We use a technique called fluorescence microscopy. It let us see multiple things at the same time on a single tumor sample. And curiously or interestingly enough, we found that two types of cells, mainly killer T cells and natural killer cells, which as their name implies are very good at killing cancer cells, If they are present in these tumors, patients have better prognosis. And importantly, where they are located is when they are good. So if killer T cells are inside the tumor, that's good. And if natural killer cells are close to the tumor, that's also good. So following this logic, we developed a scoring system. So if they are where they should be, killer T cells inside, we give 2 points. Natural killers outside, we give another 2 points. And in this way, we can classify patients in 4 groups, low, intermediate, and high. And we observed that patients that are in the high group have longer survival and also have higher survival probability. So we are now focusing on validating these results and understanding what these cells are doing in the tumors, if they are communicating with each other, and why some patients have them or not. And in the future, one possible application of this research is that these patients underwent surgery. So for example, if we apply this scoring system after surgery, it can tell us which patients we should, for example, follow up more closely, maybe closer follow-ups, or they will require a different treatment approach. But this is what we have been doing with our research. Thank you for your attention.
Thank you, Jorge. It's so great to see research like this in such a fast-growing population for cancer itself. Our next presenter today is going to be Carla Riales. She's a PhD candidate in Stephen Jones Lab at the University of British Columbia and has Canada's Michael Smith Genome Science Centre. Today, Carla will be talking to us about her work using natural language processing to facilitate precision oncology.
Hi, thanks for having me. Precision oncology is the practice of tailoring cancer treatment to the specific molecular features of a patient's tumor. In this approach, a tumor sample is collected and DNA and RNA are sequenced in the laboratory. Computational methods are then used to identify variants, meaning mutations or other molecular changes present in the tumor. To determine what these variants mean for a patient's prognosis, treatment options, or the biology of their cancer, they are matched to a cancer knowledge base, which stores structured information about what is known for each variant. One widely used knowledge base is CIVIC. CIVIC links specific genetic variants to evidence from scientific articles, summarizes how these variants relate to drug response or clinical outcomes. For example, an entry may state that a particular mutation is associated with sensitivity or resistance to a therapy. This information directly informs clinical decision-making. However, maintaining these knowledge bases requires substantial manual effort. CIVIC relies on crowdsourcing, allowing curators to submit new findings as they are published. Each submission must then be reviewed by an expert curator, who read the scientific articles and determine whether the evidence truly supports the claim. This review process has become a bottleneck. The scientific literature continues to grow rapidly, and new submissions exceed expert review capacity. Although thousands of users can contribute content, fewer than 50 expert reviewers worldwide are responsible for validating it. To address this challenge, we developed a dataset to support automated verification of cancer variant interpretations using natural language processing, a form of artificial intelligence that enables computers to analyze written text. Our dataset was curated by 13 expert curators and links more than 1,100 real CIVIC entries to sentence-level evidence from over 500 scientific articles. For each claim, we identified the exact sentences in each article that support or refute it. This provides a benchmark for testing whether AI systems can accurately assess scientific evidence. These AI models are not designed to replace experts. Instead, they act as a fact-checking layer that flags claims requiring closer review. In practice, this system has already helped identify and correct hundreds of CIVIC entries, including outdated or incorrect interpretations. By guiding expert attention towards the claims most likely to require closer scrutiny, this approach supports more scalable and reliable curation of cancer knowledge. Ultimately, it helps ensure that precision oncology decisions are based on trustworthy evidence that has been carefully and consistently verified. Thank you.
Thanks, Kara. Exciting to see how AI and humans are coming together to improve treatments. Our next presenter, Cindy Zhang, is a PhD candidate in Lincoln Stein's lab in the Department of Molecular Genetics at the University of Toronto and will be telling us about her work using AI to decode tumor origins.
Thank you so much for the introduction. I would like everyone to imagine a crime scene. Even when we don't know where the suspect is, fingerprints can still be found. And if you know how to read them, they can tell you a lot. Cancer works in a very similar way. Cancer cells leave tiny pieces of DNA in the blood that can tell us where the cancer started. That's the idea behind our work. We ask the question, can we read these DNA clues from the blood and use them to figure out what kind of cancer a patient has? This matters because today, doctors usually need a tissue biopsy to find where a cancer started, which means removing part of the tumor and examine it under a microscope. Knowing where a cancer started helps doctors choose the most appropriate treatment, but this isn't always possible. Some patients have more than one tumor. Sometimes different type of cancer can look very similar under the microscope, and some patients can't safely have surgery at all. Our team built an AI program called Deep Tumor that learns what DNA from different cancers look like. Using tumor samples, it could already identify 24 common cancer types. My project was to see if the same approach could work using blood samples instead. This is challenging because tumor DNA is only a tiny fraction of DNA found in blood, mixed in with DNA from healthy cells. Because we don't have many large collections of blood samples, we first trained the deep tumor using more tumor samples. This allows us to expand deep tumor to 29 cancer types, and it gave the right answer about 9 out of 10 times. I then give deep tumor a harder task by training it with only part of each patient's DNA information to mimic what blood sample looks like, where the tumor signal is much weaker. Even when we only provide 25% of mutation information, deep tumor was still correct in about 3 out of 4 cases. Finally, we tested it on real blood sample from 170 pancreatic cancer patients, and deep tumor correctly identified the cancer type in about 3/4 of patients. These results are very encouraging. They suggest that a blood test like this could potentially help doctors when tissue samples aren't available or when it's unclear where the cancer started. For patients, that could mean faster answer, fewer medical procedures, and more personalized care. And our next step is to collect more real blood sample and use them to further improve the tool. This is just one small step, but we believe it's a meaningful step towards making cancer diagnosis more accessible and less invasive. Thank you so much for your attention.
Thank you so much, Cindy. It's so interesting how we're starting to use AI to help patients like us go through less of the invasive procedures and possibly get faster results.
Thank you so much for all these fantastic presentations. And I will give everyone a big congratulations for staying on time as well. So thank you. We will now open it up to questions. As I said at the beginning, if you have a question, please, you can either type it in the Q&A box, which you can find at the top of your screen, or you can raise your hand and we'll allow you to unmute yourself and you can ask your question live. So Jen, I think you have a question to get us started.
Yeah, Andrew, I have a question for you if you don't mind. Given your findings on why bladder tumor cells mutate, I'm just wondering, is there a way that we could possibly screen for the low oxygen cells and then be able to maybe use an oxygen-based therapy, maybe like a bariatric type chamber, to help reoxygenate them so they don't mutate?
Yeah, what a great question. Thank you for that. So first, for detection is actually easier. So when we remove the tumor from these patients, they get treated with a turbate. So their initial primary tumor is resected. And so we have that tumor section. And so we can test that section for its hypoxia. So how deprived of oxygen is that section? And so from there, the hope is that you could tailor treatments. And so it wouldn't be a systemic treatment necessarily like a bariatric chamber, but there are drugs that can target specifically these pathways. So less about the deprivation of oxygen, but more on their response. So the cellular response to the lack of oxygen. Those pathways can be targeted. And so the goal would be that you could treat these patients kind of in combination. They would get one of these, I guess, oxygen-deprived type drugs in combination with their current drug regimen. And so the goal would be that we can keep them in these early stages and easy to treat cancers. And then in doing so, we can treat that cancer effectively. And so great question. Thank you for that. But yeah, that's the goal. We could still analyze their tumor and then we could treat them effectively in that way.
Great. Thank you so much.
Thank you.
Thank you, Jen and Andrew. Sherry, you should be able to unmute yourself now. I know I saw your comment in the chat, but just give a second. See if it works. Okay, maybe we can go to Dora and we'll come back to you, Sherry. Dora, are you able to unmute yourself?
Yes, I am. Thank you, Veronique. And I just want to congratulate everybody on their work. This is exciting. I'm also a member of the patient working group and one of the co-leads of the group itself. So, and also a scientist. So it's nice to see the work that's happening and progressing some of these treatments and outcomes. And also as young generation scientists, it's nice to see that you are bringing the hope for the future as well. So congratulations to you all. I did have just a quick question for Yuanchang about his work on the circulating DNA in the various stages of progression. Do you, in addition to seeing increases in the amount of DNA circulating from the tumors, do you see changes in the signature of the DNA itself? And that could also predict some of the potential outcomes.
Exactly. That's a very good question. So in these slides, I only show the ctDNA level. So basically, that's based off a algorithm I developed by combining all the signatures into just one single score. But to answer your question, yes. So we also have the blood samples from the healthy donors, and then we did compare a bunch of the signatures. For example, the number of the mutations we detected in the blood, the fragment size, because basically the ctDNA, they tend to have shorter fragments than the normal DNA fragments in the blood. So yes, and also we also observed that the number of the mutations we detect in the blood actually increase with the clinical stages. So like the late stage you have, for example, the bigger tumor you have, you tend to have like more distinct signatures compared with the early stage or compared with the healthy donors. So yeah.
Thank you so much.
Thank you for that. Jenny, you should be able, oh, Sherry, we answered your question. That's great. Jenny, you should be able to unmute yourself.
Also, kudos to everyone presenting for making this in plain language, some great analogies that help us to understand where your research is focused and how to understand that. Thank you for that. Yuanchang, I have a question for you also about circulating tumor DNA. Is there hope or promise for this research to identify some of the harder to identify cancers that are a bit more elusive? Ovarian cancer. I got a surprise ovarian cancer diagnosis myself when I had surgery for endometrial cancer. So that's kind of my focus there on my question for you.
Thank you, Jenny. I do believe that there's hope in liquid biopsies. So the data I'm presenting today is only focused on the plasma whole genome sequencing. It's one of the technologies that we use to identify the ctDNA in the blood. So there are like tons of technology. For example, we can use cell-free RNA to look at the RNA derived from the tumor. So going back to my technology, so the reason why we are trying to use the whole genome sequencing is we are trying to target the entire breadth of the human genome. So in a traditional way, people only design a panel to target like a specific region of the human genome. So the downside of that is if in the regions you are trying to target, there's no mutations in the blood, no matter how deep you sequence the sequencing, you wouldn't be able to capture the ctDNA signals. That's why we're trying to go a very like wide breadth into the entire human genome. And then many studies have found that by targeting the entire genome, we can have a very high sensitivity in detecting the ctDNA level. Basically, the challenge right now is in detecting the early stage of the tumors. So basically, for early stage tumors, they are very tiny. So they shed a very small amount of the ctDNA into the blood. So and then we do see there's a improvement of the technology to capture ctDNA, and then but right now I can't guarantee we have 100% chance to capture all the tumor signal. But I do believe with the improvement in the technology, in the future we will be able to have a very, very high sensitivity. Yes.
Thanks. That's great.
Thank you for that. I have a related question for Cindy, actually. I wonder if, I think you mentioned you trained your tool on 19, if I remember correctly, cancer types. I wonder if you might have, if it might be difficult to detect like the origin of more rare cancer types if you have fewer samples to train on, and how, if so, how you think that might be overcome.
Yeah, sorry. So thank you so much for the question. So the question is if for rare tumors that with less sample sizes, you're wondering if it's going to be more difficult to train my model on. That's definitely a very very good question, and the answer is yes, because machine learning tools, especially AI-based tools, heavily relies on the number of samples, and because right now the number of plasma whole genome sequencing samples are not largely available yet, it becomes one of the major challenges of my project. But as you can see, a lot of people's being actively working on this area. Yuanchang's working on pancreatic cancers, and I am hopeful that and our team are also collecting more samples. So I'm very hopeful that once the data are there will be a lot of like more interesting project and tools can be developed.
Okay, thank you. With our recent announcement that we've reached the 15,000 cases for the gold cohort, maybe that gives another opportunity to look at new cancer types that you haven't been able to look at so far.
Yeah, exactly. I'm very excited about the resources provided by the network.
That's great. It's always good to see different ways that it could be applied. Thank you so much. Yuka, you should be able to unmute yourself.
Thank you. I have a question for Julia about synthetic lethality. I was curious how you were predicting the synthetic lethal interactions. It's really cool that you're able to find something where one can be drugged, but I was curious about what your approach was.
Hi, Yuka. Thank you for your question. So yes, this is something I didn't speak on during my presentation. We essentially curated a list ourselves by using gene essentiality scores from a public database called DepMap, which essentially what they do is they knock out individual genes within cancer cells and they see what the effect is on cell viability, or essentially if cells live or die after that knockout. And then we cross-validated or we sort of referenced our list with public databases of other works that have been done by other groups that have been shared publicly. And then we use that sort of refined list to identify the pairs that we would search for. I think in the future, something we're interested in doing is to see if we can discover new pairs within patient cohorts, but that would involve a lot of sort of curation of both patient data, but then as well as patient sort of outcomes. So that's a future step, a future aim that we have. But that's essentially how we identified our synthetic lethal gene pairs, and then we evaluated it with gene expression to identify if those pairs are present in patient samples.
Okay. Thank you. Yuka, do you have a follow-up to that? Or? Great. Awesome. Thank you. Isabel.
I'll follow up on a question for Julia. So there were any context dependent in the findings that you have in terms of cell type? You mentioned that you looked at different cohorts in the data set. So were there any differences between the different cancer types, for instance?
Yeah, something we realized is that the presence and sort of the protectiveness of these synthetically lethal gene pairs are very dependent on tumor types. In the discovery alone, we didn't keep that in mind in our pipeline, but when we went down to analyze its effect on patient outcomes, that is something that's really important to consider. And then also when we're doing our validation within cell lines, we did find differences in sort of the pair ability to be associated with treatment outcome to be really dependent on the cancer type or the sort of the cell type of the cell. Thank you.
Thank you so much. I'll keep going as far as we don't have hands up or questions in the Q&A, but everyone, please feel free if you do have a question to either write it out or put your hand up and ask it. I have a question for Jorge. So if I understood your slide correctly, it looked like the tumors that had both killer T cells and natural killer cells, patients with those tumors did better in terms of survival, but that tumors that had either or did similarly to tumors who had none at all. And I wonder, I know you mentioned at the end that it's sort of a future direction to look at whether these cells talk to each other, but I'm wondering if you think that there's some sort of interaction going on there that's necessary for whatever's creating better survival.
Yes. So there's like multiple routes. It's either they are doing something different, and that's why there is the survival, or the tumor itself is different, and that's why they are being attracted to it. So we are adding a layer with a different subset of patients. We are adding a layer of sequencing too. So we will get to see, for example, mutations in the tumor. Are these tumors different? We're also validating with more patients. We don't have a lot of samples, so we're trying to do that. And also we'll do, we have samples to perform flow cytometry. So you get to see in more detail what those cells are, like, for example, what kind of markers are they expressing? Is it like something functional? So it's kind of like chicken and egg. We don't know if it's like because of the cells, is that what makes it different, or because of like the tumor itself is what it's attracting these cells? But it seems also they are highly correlated with each other. So I think I think we have more cases of both of them are present than cases of one or the other. So that may also point out of like something biologically different in those samples.
Right. That's fascinating. It's really interesting to see where this project is going to go. Thank you. Yeah. I have a related question for Emma. So you talked a little bit about how the different immune activation in the V600E versus non-V600E tumors might be different. And you talked a little bit about what that might mean for treatments. Do you think it also impacts how the tumors behave and how aggressive they are? And does it impact, I guess, sort of this in the same way as Jorge just said, we don't know what comes first, but yeah, just wondering your thoughts on that, Emma.
Yeah, absolutely. I definitely think that it impacts how the tumor is behaving because I saw it in the gene signatures. So one part that we don't fully know, because this is from a public database, is they sequence the tumor, and so they have to take a chunk out of the tumor, but there might be some immune cells in that tumor chunk that gets removed. And so we don't know if the signature that popped up is from the cancer cells themselves or from the surrounding cells. And so that's a big part of validating this research. But essentially, if it is coming from the tumor cells, then absolutely the surrounding cells are influencing how the tumor is behaving.
Thank you. I'll I'll keep going. I'll ask a question to Kara next. Kara, I'm wondering if you mentioned that you found a few pieces of, I guess, questionable information that were flagged with your tool and that could then be followed up on and corrected. I wonder if during your training you looked at false negatives at all. Do you have an idea of how many pieces of information you might have that might be questionable, but that your tool didn't catch?
Yeah, so that's actually a really good question. We've gone through a random review. So we randomly selected about 100 pieces of evidence, and then we've been reviewing them as a group. We're still calculating some of those numbers because one of the outcomes of that was that we found there were areas of uncertainty where curators didn't necessarily agree on the best way to curate something. So for example, when you have evidence and it's from a cell line, when you're trying to represent what disease that should be linked to, what cancer type in the database, there's some argument saying whether it should be the cell line it was derived from, or if there are modifications made to the cell line, should it be more representative of another disease type. But overall, yes, we did measure those. I don't have hard numbers for you off the top of my head, unfortunately.
That's all right. Thank you. Yeah, I guess it's not an easy yes or no, this is correct or this is incorrect. There's a lot of nuance there.
Yeah, usually the things that pop up. Oh, sorry, go ahead.
No, go ahead.
Oh, I was gonna say usually the things that pop up as false negatives, for example, for us will be things that someone will have entered, but they're only available in like an image. So we think that with multimodal models going forward, where they can combine images and text, we might actually be able to reduce that even more.
Right. Okay. That's great. Thank you. Lincoln, you should be able to unmute yourself.
Yeah, Kara, this is also a question for Carolyn. You're not off the hook yet. I really found your presentation really very interesting. And I wonder if you had plans to use LLMs to help search and find high priority papers for curation, because that's often the bottleneck, is actually just finding the relevant papers.
Yeah, so actually one of my predecessors in my lab did a project on that. And it's called CivicMine. And it's been used to help prioritize papers. Right now, our biggest bottleneck is actually the review, though, because we have more than half the content in Civic is currently pending review, which is a really big problem. So my focus has been on the other end of it in fact checking it. But in a perfect future, we could even put them end to end, have one pipeline inputting papers that we need to curate, people curating, and then fact checking on the other end.
Okay, so actually increasing the number of unverified variants going into the process is going to make it worse for now.
Well, I would say it makes it more critical, but
Challenge.
It's always better to have more stuff curated.
Thank you.
Thank you. Jenny.
Hi. Oh, I'm blocked my camera just in case. I just want to say that, as a patient, as someone, I love science. I read about it, but I'm on that recipient end. I just want to say that it's so evident here from the selection of papers and the work that everyone's doing that it really is a team effort. What the technology can bring for one area of cancer, and the language. I mean, Kara, I love that area of research, making things a bit more simplified, even in the background that we don't even know is going on. So I just want to say kudos again to all for the curiosity, and keep it going. And it really does impact us as patients, and it is a team. And with patients being on the team too, we see what you're doing for the benefit of everybody. So thanks. I just want to thank you. No question, just a comment.
Well, thank you, Jenny. I think that's a fantastic way to wrap up this seminar today. I wholeheartedly agree, and thank you for putting it so eloquently. So thank you to all of our speakers today. Your presentations were fantastic, and thank you for staying on for questions. Your answers were also great, so we really appreciate it. Thank you also to the patient working group members who participated in this round of this new event, and especially to Sally and Jen for joining me today, co-chairing this seminar. And finally, thank you to everyone who attended. We had a really good turnout today, and I hope you all found it as interesting and enriching as we did. So keep an eye out for emails for more of these events. And thanks again to everyone. Have a great rest of your day.